So looking at our SBW, going over protein synthesis, a couple things that we have to remember is that we have DNA. And that DNA has to be turned into a different version because it cannot get out of the nucleus. So it has to be made into what we call as mRNA. That mRNA is a code that is going to make what we have, or what we know as an amino acid. And then that amino acid is then going to become this thing that we refer to as a protein. Okay, so this is the process that we're going to look at. Now, as we're looking at this whole entire process, I do want to make sure that you understand that we have a couple other pieces to look at. So we have this thing that's called a codon chart. So basically a codon chart is how we figure out what we have. So we have this DNA code. That DNA code becomes messenger RNA, and then it'll become an amino acid. Well, how do we know that? So this is the complementary to a GGT code. This is complementary to an ATC. How do I know that? Because in DNA, an A is paired with T, a C is paired with G, but when we go into A or RNA, the A goes to a U. So wherever I see a U, I know that it pairs back with an A. Okay, so on and so forth. So this code gets made into a different code, and then we have to figure out what the amino acid is. Well, in order to do that, we look at this chart. So this chart, I'll make it a little bit bigger. There we go. If I have a C, I go to this big C right here. We always start, if you look at it, the first letter comes in the very middle. So this in here is that first letter. And then we move outwards. So we would then move outwards and say, okay, the next letter is another C, so that means a C, and the last letter is an A, that means an A. So the code of CCA is going to give me the amino acid with the symbol PRO. This is literally just how the body dissects and decodes that message. So the next message or code is UAG. So we do the same thing. We go U, A, and G. In this case, it is a stop symbol. So this is where the cell would stop reading it, stop building. It's like, oh, stop, we don't have to do it anymore. So for this, I'm just going to go right there, and we're just going to continue on. So I'm looking at this code. Now I have a ACG. So I would be an A, a C, and a G. That gives me the code or the amino acid of THR. And we just keep going on down, GUA. So I have a G, a U, and an A. Gives me the symbol VAL. So we keep going until we get to another stop. So you're like, Miss Hughes, what does it have to do with anything? Well, when we're looking at DNA, remember this. DNA carries a very specific set of instructions. That DNA has the code that's in our body, but it cannot get out. So what it does, it has to get that code out, and the only way to do that is through protein synthesis, and this is that process. Because these proteins are essential for every single thing of our life. Okay, that's why they are extremely important. So what happens is it has to unzip the DNA. So it goes from having this side attached to this side, because remember, it is a double helix. It has to literally unzip the middle. So where you have two pieces that are exposed. So what it does is it only exposes one piece. And then that piece is able to be moved out. So it brings it out. So that DNA code has to be broken up. So this is how it does it. So the DNA is opened up. Then you have this thing called RNA polymerase comes in and basically reads it. It's this big giant globby thing. It says, okay, I've unzipped the DNA. Here is my half a strand of DNA of the A, T, C, G, C, A, T, C, whatever. That's my DNA. It has to then be read. So what it does is it reads it, and then it builds messenger RNA. So it goes through and it reads it and it says, okay, well, you have an A, That matches with the U in my language. A T matches with an A, matches with a G, C, G, U, A, and G. And now this is the RNA in the form of messenger RNA. So it literally goes in and it makes its own thing. It goes and creates a new language, a new code. That code is able to leave the nucleus. And that's what we need. We need it to leave the nucleus. And that's where we get into, that's the transcription part, okay? It's written into a new language. And then translation is where we put it into and we dissect it and we see what it says. So we take the mRNA, it is then read, okay? That code is read, and it reads it in groups of 3. It's supposed to be of, not af. Typo misuse. It reads it in groups of 3. So read in, we'll say groups of 3, which is called a codon. Okay, and once it reads that, we have this thing called tRNA, which is basically like a walkie-talkie person, whatever, and it looks at what the code has and it brings in the correct matching pair. So it's like, oh, you have the code UAC. Oh, guess what? I'm going to bring in the matching pair, and that brings in an amino acid. So when we look at the whole idea of building a necklace, okay, a candy necklace, mind you, your DNA has, it's the instructions. I can't put on, okay, there we go. It's in, but we need to take those word instructions and put into a different language. We're going to say those are going to be picture instructions. Because if you're like me, I need picture instructions, okay? Then I take that picture instructions and then I build whatever it is. That's the amino acid. That's when we actually build using those picture instructions. And then we get our final product. Whoops, I don't know why I said product, because I said product, which is going to be our protein. That is our finished work. Or in this case, a protein does a certain function. So that's the thing about it, is that when we look at these proteins, the shape determines the function. Just like a key is specifically shaped to fit into a lock. So if we look at our SBW, that's basically all we have, or all we're looking at is, okay, what is this process? How do we get there? And then what in the world's going on? So the first thing is, talks about protein synthesis. Why is it important? Okay. Second thing, we need to talk about how does DNA turn into RNA and then turn into a protein. And then the third is look at what's happening in transcription and translation. So let's go ahead and look at this. I am going to color code it just so that way it makes it a little bit easier. Okay, so when we're looking at why protein synthesis is so important, the biggest thing we need to understand is that the proteins are like the workers of a cell. They are doing all sorts of jobs. Okay, and if we scroll all the way down to the bottom of the article, it tells us a little bit more about it, because it talks about the process of this making proteins. It's what is used to carry out the essential functions of life. Your body has to have proteins to function. Okay? They are literally the workers that tell your body what to do. Now, if we look at how DNA becomes RNA and how RNA becomes proteins, that's through this process of transcription and translation. Okay? So you can actually use that all together. Now, if I want to talk about it in a roundabout way, we know that DNA turns into messenger RNA. We just talked about that. Turns into an amino acid. And then that becomes a protein. It does this strictly because of this process of transcription and translation. So with transcription, it takes place in the nucleus, right? It takes DNA, copies it, and it copies it into messenger or mRNA. That literally is a photocopy of the genetic information, and then it is allowed to leave the nucleus and go to the cytoplasm. That's transcription all by itself. Translation is that next step. What it does is it is in the cytoplasm, so literally it goes from the nucleus into the cytoplasm. But what happens is the ribosome will then read that mRNA code. Remember, up here's that mRNA code. And then it uses that code to assemble amino acids in the correct order. How does it do that? Your tRNA looks at the code, and then it brings in the correct amino acid. It starts to link them together, and then what happens is it forms a long chain called a polypeptide. So literally, that's all this process is about. Okay, your DNA turns into messenger RNA. That RNA becomes an amino acid. That amino acid becomes a protein, which that is a polypeptide chain, is a protein. And that's it. Okay, now as far as going and looking at sentence starters, you can use these sentence starters, just talking about how it happens. You do not need to have any quotes or anything along those lines. Okay, if you have any questions, let me know.